So in the last lecture, we talked about the first and second lines of defense, innate immunity and inflammation. Chapter 7 actually focuses on then adaptive immunity, the third line of defense. We talked about it in the beginning as it's acquired or specific. It comes through slower and it has a very specific process. It has particular invaders and diseased tissues that it targets, and it has the sole purpose of getting rid of them. In order for our adaptive immunity to kick in, our first line of defense or our external barriers have been compromised and inflammation has already been activated. Adaptive immunity will slowly come in to help the initial defenses of the first and second line of immunity to fight infection. It also begins the process against reinfection. So, how is adaptive immunity different from inflammation? Well, we've already mentioned one, it's slow, whereas innate immunity and inflammation is rapid and immediate. Adaptive immunity is also inducible, which means that it has the effectors of the immune response, like lymphocytes and antibodies. They're not already there in large number, but are rather produced in response to the pathogen. This is why it takes longer. So let's look at the elements of adaptive immunity. First, we have antigens. These are the targets of the adaptive immunity heroes, which are antibodies and lymphocytes. Antigens are small molecules that travel with proteins, carbohydrates, or lipids. They're found on the surface of microbes, in infected cells, or on abnormal tissues. When we're being formed in utero, lymphocytes undergo extensive differentiation, or in simple terms, they become specialized. Some of these lymphoid stem cells will enter the thymus and become T cells, while others enter certain regions of bone marrow and become B cells. We also call these T lymphocytes and B lymphocytes. Surface proteins on the cells are going to indicate if they're a T or a B. Both of these cells are going to develop antigen receptors on the surface. What's specialized about these receptors is that they're programmed to recognize only one specific antigen. When we are born, our B and T cells are already capable of recognizing millions of antigens. Having these T and B cells lined up at birth is called generation of clonal diversity, referring to the millions of antigens that the receptors of the B and T cells are programmed to find. As the B and T cells leave the bone marrow or the thymus, they are known as immunocompetent, which means that they now can respond to an antigen that they may find. They are new to this. They have never found an antigen before. So they are naive. These cells are going to enter the blood and the lymphatic systems and migrate to the lymph nodes or the spleen, which are also known as the secondary lymphoid organs. Some of the B and T cells may stay in the organs and others will re-enter circulation. About 70% of the circulating lymphocytes will be T cells and approximately 20% will be the B cells. These circulating antibodies are also known as our humoral immunity. They will circulate and find microbes and microbial toxins. When they do, their receptors will bind to the antigens and the bacteria and the viruses. T cells that are circulating defend particularly against viruses and abnormal cells like cancer cells. This is known as cell-mediated or cellular immunity. As the last part of the introduction to adaptive immunity, we need to talk about active and passive. Active immunity or active acquired immunity is what develops in response to an antigen. An example of this would be fighting off a cold. In passive immunity or passive acquired immunity, this is when pre-formed antibodies or lymphocytes is administered to a person. An example would be a mother giving antibodies to their child through breast milk. There's a big difference between these two. Active immunity is pretty long-lived, and passive immunity is only temporary because the donor's antibodies or the T cells are eventually destroyed. Let's do a quick question here. Which of the following terms describes the types of immunity that occurs when pre-formed antibodies transfer from donor to recipient? As we said on the last slide, when it is pre-formed and it is delivered to the recipient, that is passive immunity. So let's talk more about antigens or the bad guys. We tend to use antigen and immunogen interchangeably, but they do have some differences. Typically, an antigen is describing a molecule that binds with the antibodies at the antigen receptors of the B and T cells. An immunogen, though, specifically binds with receptors and will induce the immune response. So it would make sense then that all immunogens are antigens because they bind at receptors, but not all antigens are immunogens. Sometimes an antigen will not produce an immune response. So what are common antigens? Infectious agents, allergens, chemical agents like medications, abnormal molecules that are found on the surface cells like infected cells or cancer cells. With antigens, the size of them usually correlates with its ability to induce an immune response. If it is a larger molecule like a protein or a polysaccharide, they are considered the most likely to produce an immune response. On the flip side of this, we have haptens. These are low molecular weight molecules. They are too small to induce an immune response by themselves, but can join with larger molecules that will act as a carrier and help produce an immune response. An example of a hapten would be the oily sap that is found on poison ivy. On its own, it doesn't produce an immune response, but when it comes in contact with the skin, it's going to bind with the larger proteins of the skin and become immunogenic or produce an immune response. It is a hapten. Now we're going to talk about how B and T cells work together. We mentioned clonal diversity a little while ago. This is when the millions of antigen receptors are formed on B and T cells in utero and throughout our life. They have the capacity to recognize almost any foreign antigen that is found in the environment. Again, in utero, the stem cells move to the primary lymphoid organs of the bone marrow and the thymus. The bone marrow produces B cells, and the thymus produces T cells. They are immunocompetent and naive, and they have the ability to react with antigens. Some go in circulation, and others hang out in the spleen or the lymph nodes, which again are secondary lymphoids. Diving a little bit deeper on B cell development or lymphocytes that come from the bone marrow, what makes a B cell a B cell? It has a specialized receptor or a BCR. As previously mentioned, the receptor is on the cell surface and communicates intracellularly. The role of the BCR is to recognize the antigen and communicate the information to the cell's nucleus. So in the first class, we talked about DNA, how it can get rearranged through recombinations. Frequently, somatic recombination or rearrangement can result in a BCR that recognizes and attacks its individual's own antigens, an inadvertent attack on self. We see this in autoimmune diseases and hypersensitivities. Many of these bad B cells are eliminated in the bone marrow. This is referred to as clonal deletion or central tolerance. They're usually eliminated through apoptosis. The body knows that they're not good. Over 90% of B cells are going to be eliminated because of this. T cell development, as we know, is when stem cells are differentiated or made unique in the thymus. These are unique because they are under the influence of hormones in the thymus and the cytokine IL-7. They also have gained their TCRs or T cell receptors to fight a diversity of antigens. Central tolerance or clonal deletion can also happen with T cells. 95% of developing T cells are also clonally deleted because of their potential autoreactivity. Clonal selection then is referred to as a symphony of cellular reactions, and it is the second phase of the immune response. This is when the antigen gets processed and there's a specific immune response. So know here that the antigen initiates the process of clonal selection. There's a complex interaction here that among the cells in the spleen and the lymph. Clonal selection begins at birth and continues through life as new antigens are introduced to the body. The process of clonal selection is then initiated when B and T cells interact with the antigen. The antigen must be processed. It cannot react directly with the cells unless it is shown or presented in a specific manner. This is the job of the APCs, or antigen processing cells. These cells are usually made up of B cells, macrophage, and dendritic cells. So how this works is that the antigens enter the body, circulate in the blood or lymph, and will undergo phagocytosis and destruction by the antigen-presenting cells, the APCs. These are referred to as exogenous antigens. There are also endogenous antigens, like abnormal cells due to injury, viruses, mutation of DNA, like a cancer cell. Regardless if they originate from inside or outside of the body, both types of antigens undergo processing and presentation. So this gets really into depth, but think of processing and presentation as winning a fight and getting a trophy to show your friends. Exogenous and endogenous antigens are cut up by enzymes into small fragments that can no longer cause injury because the cell recognizes them. And then they're presented to specific types of T cells by those antigen presenting cells. It's a look what I got type of interaction. TC cells, that's uppercase T, lowercase c, recognize intracellular or endogenous antigens that are presented by the MHC class 1 molecules on the APCs. MHC is basically the presenting molecule. As a side note, MHC is our major histocompatibility complex, is a glycoprotein found on all cells except for red blood cells. The MHC class is determined by its molecular makeup. TH cells, that's uppercase T, lowercase h, recognize exogenous antigens that are presented by the MHC class 2 molecules on the APC cells. There's a really good video that I posted in Eagle to help you understand this concept. Here's a quick question. Urushiol, a toxin found on poison ivy, is an example of an antigen that does not produce an immune reaction. True or false? The answer is A, true. Remember I said that this is a hapten. By itself, it is not something that induces an immune response. But when it hits the skin and it uses the protein as a carrier molecule, then it becomes immunogenic. The second step of clonal selection is the collaboration between cells, resulting in the production of effector cells and memory cells. Effector cells are made up of plasma cells, and the cells we just talked about, the TH and the TC cells. There are three signaling events that need to happen within these collaborations. The first is the antigen recognition at the receptor of the T or B cell, the activation of internal cellular communication, and the response of two specific cytokines. If one signaling event is missing, an immune response will not be produced. T helper lymphocytes. We have talked about the TH lymphocytes a few slides back. These are lymphocytes that recognize exogenous antigens and are presented by the MH class 2 molecules on the antigen presenting cells. But before the cell is activated, a few things need to happen first. First, the antigen has to be recognized and bound at the receptor of the TH cell. The binding is then strengthened by a protein known as CD4. These proteins will interact with CD3, which are signaling molecules, and activate intracellular signaling. With this communication, the TH cell starts to activate. Interleukins 1 and 2 provide additional signals, causing the TH cell to mature into a functional helper cell. Of course, then, there are different types of TH cells. TH1 cells help with TC cells. Th2 cells help develop B cells and therefore humoral immunity. Th17 cells secrete lymphokine that activate macrophage. And Treg or T regulatory cells limit the immune response. In the case of superantigens, some viruses and bacteria can disrupt the interaction between the antigen presenting cells and the T helper cells. In essence, they can bind to other receptors that are not theirs. Therefore, they activate a large population of T helper cells and bring on large amounts of cytokines. These inflammatory cytokines usually result in a systemic reaction. It can include fever, low blood pressure, and potentially shock, which could be fatal. Some examples of superantigens are the toxins that are produced by staph and strep, otherwise known to cause toxic shock syndrome and food poisoning. We have also talked about TC cells, or T cytotoxic cells. These are the cells that recognize intracellular or endogenous antigens that are presented by the MH class I molecules on the antigen presenting cell. These are usually virus infected or cancerous cells. The making of these cells requires the same type of intracellular communication that TH cells need to become mature. This is a visual, and it's on your book on page 165. So you can see in the middle is the TC cell that was made in the thymus. From the top of it, you can see its receptor, that it's connecting with the antigen that is presented in this case by an abnormal cell. You can see the CD8 protein strengthens the connection. At the same time, at the bottom, the TH or T helper cell releases cytokine, which helps with cell signaling. Ultimately, with all of this going on, the effector TC cell will be formed, and it's ready to kill other abnormal cells that are the same as the one it originally came into contact with. What is different about B cell clonal selection is that not only can they see processed and presented antigens, they can react with these antigens even though they have not been processed. B cells that have specific receptors can be stimulated. B cells that have CD21, for instance, CD21 has an affinity for opsinin. Remember, we talked about that in the complement cascade. They're proteins that can induce phagocytosis or mark things off for destruction. If this occurs, then activation of the B cell can occur without processing. B cells are also different in that they become a plasma cell. Plasma cells are essentially activated B cells that produce antibodies. These antibodies are capable of detecting antigens as well. So you can see here in the visual the B cell clonal selection. This is also on page 165 of your book. At the top left, you can see the B cell with the receptor coming from the top. The antigen is making the connection. The antigen is processed and then presented to the T helper cell with the help of the cell signaling cytokine. And then the activated B cell becomes a plasma cell that can then release antibodies. So remember B cells, antibodies. So we're still talking about cells communicating and interacting in the immune response. Memory cells are made up of both B and T cells. Memory cells are very long-lived and a part of a very large population. They remain inactive until there's an exposure of the same antigen. Know that memory cells do not require much transformation. They rapidly become new plasma cells or effector T cells without the interactions we talked about in the T and B clonal selection. So antibodies, an end product of the immune response. Know that antibodies are what responds to antigens. There are several different types or classes. There are differences in their structure and function. Here's a visual of some of the antibodies. This is on page 166 of your text. We're going to go a little bit more in depth on the difference on the next few slides. Know that the most abundant antibody is IgG. 80 to 85% of our antibodies in the blood are the IgG antibodies. They protect us from most infections. Maternal IgG will cross the placenta and protect the newborn for at least the first 6 months of life. IgA molecules are found in the blood, so know that's where they work to prevent infection. Also know that IgA are specific to bodily secretions. Secretory IgA. It's considered a dimer because it's consisting of two molecules that are held together by a J chain and a secretory piece. And you can see that in the picture in the previous slide. This secretory piece protects IgA during transportation through mucosal and epithelial cells, protecting it from enzyme degradation. Know that IgM is the largest antibody in size and typically the first antibody produced during the initial response to the antigen. This antibody is considered a pentamer because it's consisting of 5 smaller identical molecules better stabilized by a J chain. Again, IgM is the first antibody produced during the primary response to the antigen. Know that IgM is a little bit different because it's usually made in neonatal life. So if there's an infection in utero, there will be increased IgM in the body of the baby. IgD is usually in low concentration in the blood circulation, and it works as part of the B cell receptor on the surface of early B cells. Then there is IgE. Normally, IgE is low in concentration in the blood. It has very special functions as a mediator in an allergic response. It also defends us against parasitic infections, especially parasitic worms known as helminths. Know that IgE is typically the cause of allergies like hay fever, dust, and bee stings. If there's an invasion by a large multicellular parasite in the mucosal tissues, IgG, IgM, and IgA are going to bind to the surface of the parasite. Complement will then be activated and chemotactic factors are generated for neutrophil and macrophage to come. But this response does not do major damage to the parasites. Only eosinophils can adequately damage a parasite because of the special contents of its granules that we talked about in the last chapter. IgE is designed to initiate an inflammatory reaction that attracts eosinophils. That is why there are elevated levels of IgE commonly found in parasitic infections. You can remember the E in IgE matches the beginning of E in eosinophils. Ultimately, the parasite is killed when mast cell receptors bind to the IgE and the IgE binds to the antigen molecules from the parasite, ending in death. A part of B cell maturation that we didn't talk about is called a class switch. This is the process that changes antibody production from one class to another. B cells start off producing IgM and IgD, so one of these may switch to a new line of antibodies. Remember when we talked about plasma cells coming from B cells and producing antibodies? Right before this, the B cell has the option of becoming a secretor of IgM or changing the class of antibody to a secreted form of IgG, IgA, or IgE. So it makes sense then that the B cells may produce a population of plasma cells that produce many different classes of antibodies against the same antigen. Let's get into the 3 parts of the antibody molecule. Antigen binding fragments, or Fab, have the ability to bind antigen as it contains receptors for the antigen. The Fc, or the crystalline fragment, is the portion responsible for most of the biological function of the antibody. And lastly, polypeptide chains. 2 of them are identically light chains and 2 of them are identically heavy chains. Non-covalent bonds hold them together. So in the picture, you can see the Fc portion towards the bottom of the antibody. Remember, this portion carries out the biological function of the antibody, what it does. At the top, you can see the Fab, which is where the receptor is located. And you can see the incoming antigen looking to connect to the receptor. Also, I want you to notate here the epitope and the paratope. The epitope is the key, which goes to the paratope, which is the lock. The paratope is on the antibody, the lock, and the epitope is usually on the antigen, the key to the lock. Antigens are relatively small, so a large molecule of protein or nucleic acid will usually contain multiple and diverse antigens. Where the antigen is recognized by the antibody, as I said in the previous slide, is the epitope. This is also on page 166 of your text. The matching Fab portion that's actually on the antibody is the antigen binding site, otherwise known as the paratope. So the epitope fits into the paratope. It's like a lock and key, and it is held together by non-covalent chemical interactions. So we talked about Fc and how it is responsible for the biological functions of the antibody. So what are those functions? While we know that their main function is to protect us against infection, this can be either directly or indirectly. When it is considered direct, it's done by the antibody alone. Know these direct effects. It does this by activating or blocking the binding of antigens to the receptor, which is called neutralization. Or they can protect us through agglutination, which clumps insoluble particles that are in suspension together. Or they work through precipitation, making a soluble antigen into an insoluble precipitate. As a side note, we know that bacteria can secrete toxins that are harmful for us. Know the examples here are tetanus or diphtheria. Most of these toxins are proteins and can do major damages to a cell. Protective antibodies that are produced against the toxin are known as antitoxins. They bind to the toxin, prevent interaction intercellularly, and neutralize the effects. So, indirect antibody functions. This is specifically through the Fc portion of the antibody. This is where inflammation, phagocytosis, and complement comes in. We talked about all of those in the last chapter. Essentially, phagocytic cells have receptors that bind to the Fc portion of the antibody, making it an opsonin that facilitates phagocytosis of bacteria. So here you can see the direct and indirect ways that an antibody works. This is on page 167 of your text. So in the first upper hand left-hand corner, you can see the antibodies are the Y shapes. They are neutralizing the virus before it hits the receptor. Or in the picture next to it, there is neutralizing of the toxin that is made by the bacteria before it hits the receptor. In indirect, the classical implement pathway is initiated on the bottom left-hand corner, and next to it is phagocytosis. Earlier in the lecture, we talked about immunocompetent lymphocytes, how they migrate among the secondary lymphoid organs, the spleen and the lymph. These all make up the systemic immune system. But another type of immune system we have is called the secretory or mucosal immune system. This protects the external surfaces of the body through our lacrimal and salivary glands. It's a network of lymph tissues that we find in the breasts, the bronchi, the intestines, and the GU tract. Plasma cells in those sites will secrete antibodies and secretions like tears, sweat, saliva, mucus, and breast milk, and that helps prevent antigens from affecting body surfaces and causing systemic disease. We find IgA to be the dominant secretory immunoglobulin, although IgM is also present. IgA prevents attachment and invasion of the pathogens through the mucous membranes in the GI, GU, and pulmonary tract. Here you can see how the lymphocytes circulate through the body in a separate pattern or immune system than other lymphocytes. These lymphocytes are from the mucosal associated lymphoid tissues, otherwise known as MULTs. The antibody immune response has been divided into two phases, a primary and secondary response. The primary immune response is characterized by the initial production of IgM, followed by the production of IgG against the same antigen. Their quantities are usually equal. It's important to note here that the amount of antibody found in a serum sample or blood sample is frequently referred to as a titer. A higher titer means more antibodies. So we measure this, we can see where we are at in the immune response. After exposure to an antigen, there's usually a latent or downtime, which is when clonal selection is happening. And after 5 to 7 days, IgM is then detected in the circulation. The secondary response is when there's an exposure to the same antigen. This is characterized by a more rapid production of a larger amount of antibodies compared to the primary response. This is a result of the memory cells working as they require less cell transitioning. Know that IgM may be produced in the secondary response, but IgG's production is increased considerably, making it the predominant antibody in a secondary immune response. Natural infections, like in the case of rubella, may produce measurable levels of IgG for the life of the person. There are some other cells that are part of the cell-mediated immunity. Natural killer cells are one of them. These are a special group of lymphoid cells that are important not only to adaptive immunity, but also innate immunity. They're similar to T cells, but they do not have receptors that are antigen specific. Instead, they have receptors that identify protein changes that are common on the surface of cells that are infected with viruses or have maybe become cancerous. When we are considering pediatric immunity, we should know that the fetus usually has sufficient IgM, but it's going to be deficient in IgG and IgA responses. Maternal IgG antibodies are transported across the placenta into the fetal blood and protect the neonate for at least the first 6 months of life, as we mentioned before. When a child is born with deficiencies in their antibody production, they are considered immunologically immature. Lastly, when we're considering the elderly and their immune system, they have a decreased thymus size, which means they have a decreased T cell activity. As the thymic hormone drops, so does the differentiation of T cells. Those who are elderly also tend to have increased level of circulating autoantibodies, which are the antibodies against self-antigens seen in autoimmune responses. Overall, they have a decreased antibody response to antigens. This puts them in risk for impaired wound healing and is usually because of chronic illnesses.